It's good evening for Dr. Bly. He's joining us from Germany and has graciously agreed to stay up quite late to give us our talk. What time is it there, Felix?
So it's just 8 here, 8 in the evening.
8 p.m.
Yeah, 8 p.m. So thanks for the invitation.
Yes. Yeah, I'm going to give it a few minutes, let people trickle in. We'll start at the top of the hour here. In the meantime, I'll let everybody know what our agenda is today. It's posted as all the agendas on the OHA website. We're going to have a few housekeeping items, mostly attendance, minutes, discussion, and approval from the last meeting, and then an introduction. And we'll hear from our guest today, Dr. Felix Bly, who's joining us. So that will be about 30 minutes. Then we'll have approximately 1 hour of Q&A, question and answer discussion between members of our product subcommittee and or full board who are in attendance today. Then at the end, we'll have 10 to 15 minutes of time for public comment. If you wish to submit a public comment, present some ideas, please send them via chat to Nick Riley by, let's see, 12:30. You'll have about 2 to 3 minutes depending on how many comments we get. As all public comment sections, any questions that you pose will be rhetorical. It's not a discussion-based Q&A format in the public comment section. And so if you can please submit your intent to comment to Nick Riley by 12:30. Okay, great. I'm seeing folks are arriving now. Hello, Angela. Hello, Mason.
Hello.
Hi, Angie. Great. So I'm going to go ahead and, hello, get started here. So first of all, attendance. I know we have some people who are joining us late today. Rachel Knox and David Hart will join us a little later on. I don't think we have any voting items, so we don't need quorum, if I'm correct there. I see Mason Marks is here. I'm here. Angela Carter's here. Is Steph Bars with us? Does anybody see Steph? Steph is not here. Okay, noted. If anyone sees Steph arrive, please notify me or Nick. All right, next item, housekeeping. So the minutes from our last meeting from June 10th from the Paul Stamets presentation, did folks have a chance to look those over? Are there any edits or suggestions for additions? I realized that I missed to note one public comment. Will McHugh commented on the value of using hyphal cultivation on mass in bioreactor format, and I will add those edits and send them to Nick, Andre, and Angie. With the exclusion of those additions, are there any other edits or comments? And if not, can I please hear a movement to approve the minutes?
Do we need a quorum to approve them? I'm not actually sure.
My understanding was the quorum is for voting. Angie or Nick, could you clarify?
Hi, everyone. I do believe that if you're voting on minutes to approve them, that's considered voting. Nick, have we had this question come up before?
Not specifically, but I would agree. I think that a quorum would be required to approve the minutes.
You can just push it off till later.
Yeah.
Yeah.
That's what we'll do. Thanks for your guidance. We'll circle back to the minutes. Okay. With that, I'll move on to the introduction of our speaker. And Dr. Bly, if you can cue up your slides while I do your introduction, please. Wonderful. There we are. Okay. So it is my pleasure to introduce our second speaker and my mycological colleague, Dr. Felix Bly. Dr. Bly is a world-renowned expert in natural product analytics and drug discovery, the CEO of Miraculix, co-founder of Mimosa Therapeutics, where he is also the vice president and director of the analytics department. Felix studied at Friedrich Schiller University, receiving his bachelor's and master's degrees in 2012 and 2015, and then continuing on for his PhD at Hans Knoll Institute in the laboratory of Dr. Dirk Hofmeister. Together, during his PhD research, they were focused on understanding indole alkaloids, including psilocybin in psilocybe species and relatives, resulting in a series of very high-impact scientific publications. Felix's papers have very long fascinated me, and it's incredibly challenging to briefly summarize his voluminous research. But of direct relevance to our work, he led colleagues in the elucidation of the biosynthetic pathway for psilocybin, publishing groundbreaking literature on in vivo, that is, in whole live mushrooms, psilocybin synthesis, and later harnessing that process to increase production rates in model organisms like yeasts and bacteria, and finally in vitro settings in lab in a test tube. He also discovered the beta-carbolines, compounds present in some psilocybin-producing fungi, which are potent inhibitors of enzymes that degrade psilocybin and related amines. In other words, the presence and abundance of this molecule class, together with psilocybin, affects the duration and intensity of psychedelic experiences. He has a very impressive CV, including winning the prestigious Phoenix Pharmacy Science Award in 2018. And his most recent work, which I hope we get to hear about today at Miraculix, includes development of a visual color change diagnostic similar to a pH dip strip that is capable of rapidly quantifying psilocybin from fungal tissues. With that, thank you very much for joining us, and welcome, Felix.
Thank you for the invitation. So I'm really proud to present here today. And so is now the time to start?
Yes, please go ahead.
So then, welcome everybody. In Germany, it's already evening. So I will just start with this nice slide with some psilocybe crystals in the back. Yeah, thanks for the introduction. So my name is Felix, and I will just show at the beginning the slide with the conflict of interest. As mentioned before, I'm the CEO and chief scientist of Miraculix, which is a university spin-off, and we are developing colorimetric test kits, and yeah, also working with Mimosa, and there we are want to produce really natural psilocybin and bioreactors. And besides that, I'm also a project manager for the colorimetric quantification of alkaloids, and at the moment we apply for a new grant. So I try to make a really short a presentation of a lot of stuff today. At first, really short introduction. I will tell you then a little bit more about the biosynthesis cluster. Next, about the in vitro synthesis of psilocybin, the beta-carbolines out of psilocybe, the extraction and the analysis, and last also something about the test kits. So let me start with the chemistry of psilocybin. So in the next slides, we'll talk a little bit deeper in this. So I want to keep everybody updated that we can talk about the same. So the psilocybin molecule I posted on the left side, it's on the chemical side, a really easy molecule, but also has some interesting features. So at first, there's an indole skeletal structure. I numbered the residues here, and as you can see on the fourth position, there's an hydroxyindole residue, and on that, a phosphate ester. And then another interesting one is the tertiary amine here. And the psilocybin, to keep that in mind, this the stable product of psilocin. So this phosphate ester needs to be cleaved, and effects do not occur until the decleavage of this phosphate ester. And with this ester, it has a pretty high melting point at over 200 degrees. So let's start with the biosynthesis cluster. In the lab, we worked with 2 different strains. We genome sequenced them. I posted them on the left side. It's Psilocybe cervica, a local strain we found here, and the pretty common Psilocybe cubensis. And we were able to find a region in the genomes where at least the genes we expected were in a really small area. I will show you here the cluster we found. So A was from Psilocybin cervica and B from Psilocybin cubensis. And there we found a region where in the middle of, let's say, 25 kb, are the kinase, a hydroxylase, a methyltransferase, and also a decarboxylase could be found. So with this information, we gone one step forward. We put this into E. coli, produced our own enzymes, purified and isolated them, and then we could really do in vitro characterization of these enzymes. And with this work, we could establish the biosynthesis. And this works with the affinity of the enzymes. So we fed them the different substrates and looked which one was really taken from the enzymes. So, and the start of this biosynthesis is done with the decarboxylase, which cleave the decarboxylation of this group here to from L-tryptophan to tryptamine. So this was the first step of the biosynthesis. The next step is an hydroxylation with the enzyme PCH, and on the fourth position of the indole ring, and then you get these 4 hydroxyindole residue. And the next step was obviously a kinase, because this is a really specific enzyme. It's so it adds phosphate ester here at this hydroxyindole residue, and then we get no biocysteine. And at the end, there's multiple methylations from an enzyme called CM, a methyltransferase, with the usage of S-adenosylmethionine. And it has a really high affinity to methylate 2 times, and that's the reason why also in extracts are mostly psilocybin is found. And at the end, we also saw that psilocin is rephosphorylated from the kinase back to psilocybin, which yeah, indeed makes a lot of sense in the fungi, because they really want to take this stable psilocybin and not the unstable variant. But I will talk about the unstable variant in the next slides. So for the in vitro synthesis I developed, I took another enzyme, which is also the same organism. So it's also from Psilocybin, and it's TRPB. And TRPB does makes with 4-hydroxyindole and L-serine, the 4-hydroxy L-tryptophan. And we found out in the experiments that the decarboxylase is really flexible also for other substrates. So we could use this enzyme with to use a really cheap 4-hydroxyindole as a precursor, fed this, and just to imagine, you can mix them now, these 4 enzymes in one pot. And the decarboxylase just accepts it as the normal substrate, and the biosynthesis goes really up to the psilocybin. So it was really an advantage to get this TRPB, because CH is kind of dependent on a redox system. In the study before, we did this in Aspergillus. So this can be now done in one pot, really. And so the good thing is it's going to be a scale up, and you can also get really clean products compared to in vivo or natural extracts. And also the good thing is that you get in the end really the stable product, the psilocybin. And to sum this a little bit up, I made the next slide a little bit about the benefits and the disadvantages of the in vitro synthesis. And for that, I just wanted to start with a short sentence out of the review I posted down here. And so it says, currently the cost to synthesize 1 gram of psilocybin, for example, for phase 3 clinical studies based on the chemical synthesis with the Hoffman protocol, to be honest, that's a really old protocol, is estimated to be greater than $2,000 US. So there are also novel techniques. I'm sure they're much cheaper, but this was the method where I could really found here a number behind 1 gram. So what's now the really advantage of the in vitro synthesis? So what is good, that the reaction can be really easily scaled up. So you can do also a bigger scale-up assay and obtain high amounts of psilocybin. It's also really good for, it's able for good manufacturing practice, GCMP. So you can control every factor and standardize it really good, and you have a really standardized product at the end. And for example, also compared with the whole cell catalysis, the products of the in vitro synthesis are much more cleaner. They have much more less background inside, and you just mostly get your product you want. But it also has some cons. For example, you need to do some efforts at the beginning to really produce and purify the enzymes. And yeah, as working with enzymes, you have problems with stability, the solubility of the enzymes, and also the supply of the co-substrates. So for example, if you working in vivo, there the organism just delivers the co-substrates. And in this case, you have to add the ATP, for example, or the S-adenosylmethionine. Okay, so next big step. I wanted to introduce this subject shortly now with these nice pictures. And we're talking about beta-carbolines I found in the psilocybin. So to look in the left corner, there you can see different fruiting bodies. From the left two are cubensis, then here's cyanoscans and also mexicana, and here the extracts. I did this with in different steps, and at the end you obtain really nice colored extracts already under normal light. But the interesting feature occurs when you look under these extracts under UV light or black light. Then you can see really nice fluorescence. And it's also interesting that different strains or different species also seems to have different fluorescence. And for this subject, I extended my PhD because I really wanted to figure out this topic, and I had the idea to, for these beta-carbolines earlier on, but as it took also some time to really convince my professor. So at the end, I found a lot of new beta-carbolines, at least new in these psilocybin fungi. And under them are some prominent ones like harmane or norharmane, but also harmine, perlulurin, or harmol. And why is this interesting now? I have made this slide. So the psilocybin on the left side produce the tryptamine, as I said, psilocybin, which is the product and is really inactive until it's consumed, and the phosphate ester are going to be cleaved off. And then the psilocin is really the psychoactive compound here. But sadly, this compound also going to be degenerated or from an enzyme called the monoamine oxidase A, so to an inactive product. And what is now the interesting feature of these beta-carbolines, for example, harmine or harmane, they inhibit the work of these monoamine oxidase A, which are, now could have a little bit more effects also on the psilocybin effects. So just want to show shortly the degradation here, which happens about the monoamine oxidase A. So you have this psilocybin, which is going to dephosphorylate it and then degrade it to 4-hydroxyindole acetaldehyde. And the same process with the same enzyme also happens with the human body serotonin, which is going to be also degraded to 5-hydroxyindole acetaldehyde. And if you see there's between serotonin and psilocin, there's really just a small difference in the 4th or the 5th position of this hydroxyindole, and the rest is nearly the same. And so this concept of mixing beta-carbolines together with a tryptamine is not a new concept for at least for the humans. And for that, I wanted to show this nice slide. So let's start on the left side. So we have the psilocybin cubensis in this case, which produce psilocybin, as mentioned, which is going to be dephosphorylated. And as mentioned, the monoamine oxidase A inhibits or degrades at least this compound, and the beta-carbolines inhibit the work of the monoamine oxidase A. And the humans also use this in a brew called ayahuasca. So at least for several hundred years, we could already found the existence of this brew, and it's a mixture of lian, the Banisteriopsis capi, which has different beta-carbolines as harmine or harmine inside, and a mix with tryptamine, the DMT, out of a plant called Psychotria viridis. And alone, the DMT is only really short of short effects, but mixed together, the effects last really over hours. So this discovery of the beta-carbolines now really ask questions: how much effects play they in the role of the effects of these fungi? To be honest, we found really just small amounts of these compounds, and so the effects should be just small, and like a shorter onsetting or maybe a prolonged effects, something in this direction. But definitely there needs to be more work done in this direction to say a clear sentence. So that's just hypothesis, if it's really plays an effect in this case, or yeah, or how big the effect is in this direction. And so to sum this a little bit up, I made this slide, sorry, about the different indole alkaloids I just found in psilocybe to show how diverse it is. And so the first I showed to you already is the biosynthesis of psilocybin, yeah, with the pathway. And the next was the integration of the TRPB, which is also capable of taking different substrates. So it's flexible of different hydroxylated indoles. And I could show this, for example, if you use instead of 4-hydroxyindole, 5-hydroxyindole, the dicarboxylase also accepts it as a substrate and just makes serotonin. So with this in vitro synthesis I showed you before, with the same set of enzymes, just taking another precursor, just feeding another precursor, you get serotonin. If you feeding 7-hydroxyindole, you even get the isomer from norbeocysteine, which is called isonorbeocysteine. So the kinase was able to phosphorylate even the isomer, but sadly the 5-hydroxy residue, it doesn't take as a substrate. And yeah, the 4-hydroxyindole reaction I showed you, which is like a kind of gateway into this biosynthesis of psilocybin. And are there other alkaloids? Yes. For example, different methylated tryptophans, also out of the L-tryptophan. And as mentioned before, different beta-carbolines, which we also suspect also coming out of L-tryptophan. So, ready with that, and now some interesting stuff. I was asked to also show a little bit my experiences with different alkaloid contents I found the last years when analyzing these fungi. And for example, on the left side, I posted how, at least in the literature, this potency is published. The literature I used is posted below. So, for example, I took this 3 fungi just for comparison. So, for example, the Psilocybin cyanescens, at least the European variant, has to be 0.2 to 0.9% of psilocybin. Psilocybin cubensis is yeah, at least in literature 0.6 to 1.1, and Psilocybin semillanceata 1 to 1.8%. So, and now on the right side, the potency I measured in reality, or at least some other lab. Yeah, I will come to this other. So, for example, let's start with the Psilocybin cyanescens. There's an interesting story. I found them in the wild and just in our own local town, and they had roughly 1% of alkaloid content of psilocybin. But one town in 50 kilometers away, they had 2%. So they were collected like in the same week. So there were just different habitat. And basically they looked the same, but had maybe mostly over double the amount. And now to the Psilocybin cubensis, where I even found so the ones I growed in the lab had like maximum of 2%, which is also double of the literature. But there were also some results in the past, for example, from this Psilocybin cup, which happened recently, and a lot of different mutants of these cubensis strains occur, and they're up to almost 3.9% in the total tryptamine content. And for the Psilocybin semilanceata, I also found a big diversity. So the potentiality also really ranged here. The table behind these values. So on the left side, the literature contents, and on the right side, my own HPLC analysis, or at least with the 3.9%. The other one I wanted to really show here in this slide. So the takeaway message from this table is that there's a high variation in this alkaloid contents, even in the nature or grown in the lab. And now I want to take a little bit into the lab, how I extract psilocybin there. And it's a really a long and a complicated process, which starts at best with the freeze drying of the fruiting bodies. So on the left corner picture, you see big Psilocybin cubensis fruiting bodies, which are freeze drying, and in this size, they sometimes need 2 or 3 days to really crack it dry. The next step is to grind them up really well, for example, using a mortar and pestle. And then there's the first extraction step with methanol. And you did this minimum 2 or 3 times to really get all of the alkaloids outside. And then all the methanol has to be evaporated again. Then you use other solvents to take it up and purify these, as you can see in this picture, also either using different layers of solvents and shake it. And at the end, you get your samples, which are clean enough to be analyzed on an HPLC. So that's kind of the process, and really shortly here. And to sum this up, together with the freeze drying, it's several days of work actually to get a nice result. And the extraction is a really critical step. And for that, I wanted to add this slide. So it's really important to the proper drying and the storage of the fruiting bodies. And so in the past, there were a lot of results published showing psilocin contents. And my colleagues were able to already publish these. That is mostly dependent on bad extraction, or at least bad drying of the fruiting bodies. So if you use proper freeze drying and a good extraction method behind, at least I, for example, just found minor amounts of psilocin. So that's the reason why I wanted to tell that it's the fungi really want to have the stable psilocybin inside the fruiting bodies because it's polymerizing. And I will show you this here in this slide. So that's really a fast process. I heard these fruiting bodies here with a scalpel blade, and you can see the bluening occurs inside seconds. I hope you all can see this video quite well.
Yes, it's sputtering a little bit, but we see the bluing. That's fascinating.
And it's already now published what's behind that. And that's I wanted to show just shortly with this picture here. So there are 2 enzymes involved in this chemical reaction. So the first one is called CP, and it just dephosphorylates psilocybin to psilocin. And then there comes a lactase called L, and at first it does dimerization, and then there are also an oligomer or at least polymerization. So different chains of connected psilocybin molecules occur, and yeah, at least that's kind of a irreversible step. So you really lose potency here, and that's maybe the take-home message. If the ballooning occurs, that's psilocybin, which is degraded and made to a blue dye, and yeah, you lose this as the active compound. At least that's the assumption from a polymerized compound, that it's not be active anymore. And I also wanted to show some pictures about the analysis of psilocybin and how which machines are used, for example, for that. So in this picture, you see UHPLC together with a mass spectrometer on the right side, and there's a nitrogen generator which you need for the mass spec. And so until now, it's the quantitative determination is really hard, and it needs a lot of scientific expertise. You need this laboratory with this expensive machines, the time, and a lot of money. So, yeah, and sometimes you have even a lot of HPLCs in your lab for different extractions or purification processes, or yeah. And how does this look like if you analyze your sample with an HPLC? That's, for example, psilocybin extract, and at yeah, roughly the retention time of 1 minute, that's the psilocybin peak. So the first panel is the signal, the UV signal you get from the HPLC, and the second panel is the signals you get from the mass spec. And I just clicked in this peak here, and you can see on the right side that the mass of 285 occurs, and that's the psilocybin. So together with a mass spec, you can even get yeah, detailed information of the weight of the compound. And but you can also use the UV chromatogram, and at least, for example, all the indole give really nice characteristic spectra. And the next step would be then to use these data. So you need to do your extract, but you also need a pure psilocybin to make a standard curve and a standard dilution. So with injecting the standard dilution, you can now calculate the peak, yeah, the area of the peak, and then in your analyzed extracts with a correlation of this standard curve, which is what is really inside your fruiting bodies. So that's how it works with HPLC. And at least for me, it was a really, it's a really long process, and I thought, yeah, it needs to be a little bit more easy maybe in the future. And yeah, I started to work out test kits for psilocybin, which are colorimetric based, and they are able for the fast quantitative determination of the psilocybin content. Right. So they are mobile and really easy to use. The example for the psilocybin kit, the results are present in 30 minutes. And you can evaluate these test kits just per eye with a color scale. We're also developing an application for the smartphone at the moment, but you can also analyze it with a spectral photometer in the lab. And how does this look like? I took this sample pics out of the lab. So on the left side, there are different biomasses which contain psilocybin from the left to the right. So Tampanensis mycelium with 1%, then with 0.3%, Cubensis mycelium with 1.6%, and truffles with 0.4%. On the right side, you can see the test vials, and based on the color, you can now already see with the color sheet we worked out the concentration of these compounds. And yeah, so it's just take 30 minutes to really look into the contents of these fungi. And I also developed these test kits for several other compounds. And in Germany, we now starting this an on-site drug checking project together in a cooperation with some drug prevention center in our local federal state. It's called the Suchthilfe, and together with the Drogerie, so we have a cooperation with a prevention center. We are doing, we were visiting parties and festivals. At least Corona is allowing this for us now, again. And so there are going to be some analysis of several compounds, and then after that, the prevention talk from really people who are experts in that, which is there for a safer harm and reduction, harm reduction purpose. Yes, and thanks. This was my short talk. I think I had roughly 30 minutes. Who wants more information can look on this website or just follow us on the social media. And now if you want, I'm open for questions.
Wow, that was incredible. So much helpful information. Thank you very much, Felix. I know I have a lot of questions, but I want to open it up to other subcommittee or board members. Questions for Felix, please.
I'm curious,
Go ahead.
without asking for any proprietary information, the alkaloids that are being tested, what is the color binding to? Does it specify psilocybin, psilocin in that class, or does it cover all alkaloids?
So, so the difference between, for example, between norbiotcysteine and psilocybin, and it's just a methyl group. And this was also the reason why I talked before about that the methyl transferase is really fine to make a double methylation. So in normal fruiting bodies, there's really just minor amounts of these other compounds present. And though that's also the reason why it's really not important. And other reasons are that recent publications really also assume that there's really no big effects from other compounds like beocysteine. There were some studies outside. So yeah, they are presented in several, just in minor amounts, most probably have no effects or even are small effects because the minor amounts. And yeah, it's really hard to, so the test kit will measure both.
Thank you.
That's a great question. And it leads to one that I have. So I was trying to do some conversions based on your data for psilocybin concentration variation. This is an idea we've been talking a lot about, how different species of psilocybin containing fungi vary in their concentrations, even mushrooms, mushroom to mushroom of the same species. So if I heard you correctly, you said Psilocybe cubensis can vary from 0.6 to 3.9% psilocybin by dry weight. So for like 3.5 grams of dried mushrooms, that's between, if I did my math right, what is that? 30 to 130 milligrams of pure psilocybin, so a 5-fold concentration difference within one species. Is that what you're saying?
I can also show, if you want, some results of this Oakland HiFi lab. I just put it in the slides behind.
That would be, yeah, it'd be very helpful. It's astounding to me how variable they are to get some numbers on this process. So literally almost orders of magnitude difference within one species.
So yeah, it's from the Psilocybin Cup 2021 from this Oakland HiFi. I just wanted to mention before showing the data. And it's a long slide, but you can, so that's our different and also famous cubensis varieties from different guys who cloned them or grow them, sent them to them, and they range from, you can see, like 0.4 up to almost 3.8% of the total content. But the ones on the top they are not also looking like cubensis anymore. So here are some pictures from these, and the winner looks like that. So that's like a coral structure, but still belongs to the cubensis.
Hey, Felix.
Yeah.
Hi, Human. How are you? I saw that before the competition. Did you do the testing on that?
No, I don't. That's was also the reason why I showed the Oakland Hi-Fi. So the table is from Oakland Hi-Fi. They posted it online, so everyone can find this data online.
Yeah, I was just questioning the testing.
So I extracted also a lot of psilocybin mushrooms, and but my highest contents was just 2.2%. But I don't have these crazy mutants tested in the lab. So I just had normal, I guess, normal strains. But at least there, the variation up to 2% is also really high, which I think that's the same evolution as you can see with the cannabis strains. They try to make it stronger and stronger, and that's just selection. And yeah, if you select them more, you will even find higher contents, I think, in the future.
Thank you for your comments. I wanted to offer a gentle reminder that public comments are welcomed via chat to Nick Riley and will be addressed at the end of our presentation. So right now we're having question and answer discussion between the subcommittee, the board, and Dr. Bly, please. Thank you. Other questions?
The image you just showed of that mushroom, which you said was a cubensis?
Yeah, it's called tidal wave.
It's fascinating. It looks like it's covered in a hypomyces. Is there anything on it? Is it just only a cubensis?
I think the black color is the psilocin. So it's so the normal fruiting bodies of cubensis grow in like 1 week mostly, and this one grows, I think, over 1 month. And so the older parts of these fruiting bodies, I think, going to be a little bit dying, and they're so the kinase cannot rephosphorylate really good the psilocin, and then the polymerization occurs. I think that also happens in petri dishes in the middle, in older petri dishes, for example. And I think the same also happens in these mushrooms here.
It's fascinating. Thank you.
That's incredible. There are, as Felix knows, fungi and Angela that look like this by nature, but this is very different from a cap and stalk mushroom that's its closest relatives look like umbrellas with a pileus and a stipe rather than like a coral or a cauliflower. So I have another question. Are the beta carbolines found in all species of fungi? Are they universally present, or does their abundance vary? If not, who has them? Any thoughts on why?
So in my publication, I think I presented the data at least of 4 or 5 species, and it was totally different, and it was also different if you look, for example, in the mycelium or fruiting bodies or sclerotia. I found even high amounts, for example, in sclerotia or in mycelium, and it was also strain specific. And at least it also seems like so I don't have really a lot of fruiting bodies out of the wild, so they just grow rarely. And at least, for example, I also just had some chances to look in them, but not enough biomass to really purify new compounds out of them. So for that, it's good to grow them in the lab. So, but that's hard for psilocybin fruiting bodies which not fruit in the lab. So, for example, if you look at cyanescens, semilanceata, all these stuff will not fruit in the lab. It's just grows outdoor, so it's yeah, you just have once a year the chance to harvest some if you find them. And so and at least that's my experiences, that in the wild collected mushrooms had much more diverse beta-carbolines, and at least that's what, yeah, and there's a lot of them, and that's not all of them. And different strains seems to be have different ones, and yeah.
So in other words, strains really vary quite a
Yeah.
bit from each other in terms of the constituents of their bioactive molecules and the ratios of those constituents to each other. So a related question then is, during your search for the Psi gene, Psi K, H, M, D, et cetera, in the biosynthetic pathway, did you come across other enzymes or biosynthetic gene clusters that you think may be active? Could you perhaps discuss the possible diversity of other bioactive molecules that we don't yet appreciate?
Oh, that's really hard to just tell from the genome. So you don't know which cluster or gene is active or not, and so that's hard to tell from a genome sequencing. So I'm not sure how the questions want to go. I'm not sure what you're looking for.
So let me clarify. So I'm thinking, given the relatively recent advances in understanding the structure and biosynthesis of psilocybin and its conversion to psilocin and the relatives and intermediates that also seem to have bioactivity, what you think the chances are that there are other molecules that have bioactivity together in these fungi that create the ensemble effect, that we don't yet know their structure, their activity, what receptors they bind to, the effect, how they're made, etc. Is that possible?
Yes, so at least I think I think they're pretty safe, as the humans consume them a lot of thousand years. But also we have to think about things like the wood larva paralysis. I think you already be aware of that. And we don't know which is really the reason for that. It could be molecules, could be just bacterial contaminations or something else. So yeah, there seems to be at least risk with these wood loving strains, but so I know what you mean, and at least it would be good to have a genome sequenced fungi to work with, and to don't have yeah, I know what you want. So even if you take them out of the nature, it would be a good idea to already look at the psilocybin cluster. If there's anything else inside or changed around, then it could be already hint for some other metabolites. But for example, for the beta carbolines, also the genes are not found. It's really hard to find the genes in the genome. So for example, for psilocybin, I did discover with my colleague, and if you know the structure of your compound, then you can guess which enzymes are involved in the process of making these. So that's much more easier if you go this way back, and then you have to also be a little bit lucky that it's clustered, and in fungi that's not has to be clustered. So there could be just 3 genes totally away from each other, but together making a biosynthesis or pathway. So based just on the genome sequence, yeah, you can run it through a pipeline, and so basically blast all this stuff and get some ideas what are the genes are. But mostly all the conserved regions are out of bacteria from the internet. So you have already these yeah, a big percentage of difference if you look into fungi, because all these also molecular tools are just developing for fungi. They're just they were made for bacteria. And if you look for these yeah, famous tools where you look for NRPR clusters or stuff like that, and so there needs to be a lot of work done in the lab to with these fungi to make it, yeah. And I think if we gain more data, and then in the future we can also make better predictions out of genome sequences. But at the moment, if you compare it and have like 50%, just, yeah, it's really hard then to tell what a gene is for.
Yes, this idea of making sense of genes and genomes, otherwise known as fungal genome annotation, is something that we think a lot about and struggle with in my lab too. So making sense of genetic data, the 23andMe for fungi, if you will, is no easy task. There's a lot of important research that needs to be done for mushroom biology. That's certainly true. I see that, let me take a brief pause here. I see that David Hart is with us. Does that constitute quorum for our voting purposes? We have, hello, David. 1, 2, 3, 4.
And I'm glad I caught your last question, Jesse. As a layman, I think I understood. I'm not 100% sure, but that was interesting. Thank you.
You're welcome. David, did you have a chance to review the minutes? We were not at quorum at the beginning of our session, and I'm wondering.
I vote to approve, assuming you already voted and you need my vote.
We didn't vote yet because we didn't have quorum. We were missing you. So if you move, maybe someone could second.
I move to approve.
I'll second.
Thank you. We're done with that. More questions, please.
Well, going back to the concentration piece on maybe a practical note, I know a lot of folks in the underground and in research have worked really hard to discover the methods by which one can optimize psilocybin production in various different species. I wonder if you have come across data or if you found information yourself in your own lab about the impacts of, I mean, insects, watering, food substrates, all of the different pieces that might have an impact on psilocybin production, so that we can sort of get a sense of how we can set limits for concentrations and, you know, have a standardization process as a part of our growing process.
So I grew really a lot of Psilocybin cubensis, and I also tried to standardize this process in the lab using the same nutrients, but still they had different alkaloid contents at the end. I think it also belongs to harvesting points and stuff like that, and the temperatures, just also how alive the culture was. I inoculated from something like that, and so my personal favor are more complex media, so I try to give a lot of diversity of nutrients, as they also find in the nature, and but yeah, it's even if you look at the same spot in the nature, so which is the same habitat and the same nutrients, they will have differences in the potency, and I think at least until we not are cleared really what are the transcription factors and really the trigger also for the production, or at least also the reason for the production of psilocybin is, then we are a little bit in the dark here, and yeah, so it's really hard and to standardize if you go them just on diverse substrates as grains, which are also diverse itself. It's much more easier if you're using if you're thinking about biology, if you're using like liquid media, which is where you can give nutrients really synthetic, so every nutrient single one and stuff like that, then this helps definitely with the standardization here.
Thank you.
More questions for Felix? I have many questions. So circling back to the piece on degradation of psilocybin to psilocin and then to this blue staining product, do you have insights from your data or the other literature into how fast this happens and what kinds of environmental factors affect that degradation rate? I've heard canonically light, heat, humidity, oxygen. Can you comment on that?
So really hardly not, but I think the problem is because there are no data outside out of that. I also looked, for example, the combination of psilocybin and other compounds which could do the same effects, but I didn't found anything really based on science, just some journeys out from people who mixed these substances before. But yeah, then it needs really to be some research done in this direction.
An area for open research. 100% agree. So building on that, if I understood you correctly, there are enzymes that convert psilocybin to psilocin, the dephosphorylation, and then also the opposite direction. And I'm wondering if you could comment on how frequently this conversion happens.
So just wanted to show the slide from that. So it's so the kinase, it's a really effective enzyme which is really fast. So for example, if you have it on your bench and feed it with the substrate, in almost 2 minutes it's totally converted and totally almost converted back to psilocybin. So this kinase is really effective, really fast, and the purpose on this is as you can think, the fungi doesn't want that it degrades inside, and really wants to avoid that, and that's the reason why this kinase is so effective. And the other enzymes called the CP, that's the total opposite of that, but that's the reaction on the injury which happens, or some other stress which occurs, which let the cells die. Like if they age, then what I told you in the petri dish, you also can see this blueing. So I always compare the psilocybin like a hand grenade, and the phosphoryl ester is like the splinter. And if you put the splinter away, then you have this really explosive reaction and the polymerization, and this polymerization is really fast. So you can see it per eye. If you work with the rotary evaporator, it already happened. It's just like a black spider net, and it's getting bigger and longer. So it's a really fast process which happens here.
Fascinating, thank you.
And it was so my colleague discovered this, and was really hard work for him, and took a long of time, because I think at the end he did some live NMRs where he let it polymerize directly NMR and looked how the signal changed and stuff like that. So a lot of people tried to analyze this blue compound, and because it's a polymer and also have different lengths of the polymer, it was so hard in the past to discover this compound. And it's really, I'm not sure why the fungi also does this, because actually it makes no so big sense, because we also didn't found the reason for the psilocybin at the moment, I think. But so maybe also it's just the psilocybin is just produced for making this blue compound, blue color.
Very interesting. Thank you.
But with like 2% of the biomass, it's really a lot. So yeah, still a big question why the fruiting bodies invest so much energy in this compound.
So one more build on that, and then I'll hand the mic off to someone else. I read in your paper that the beta carbolines are often concentrated at the growing hyphal tips. And I just wonder, given their function and this process of degradation we're talking about, if you could speculate on why there, or how these, I'm very interested in evolution, how these traits arose, and then also, how we can potentially harness them for these applied purposes.
Sorry, that's a really big, really good questions. I also would have why it's produced in the hyphal tips. But these were a nice picture from the MALDI imaging you saw, I think. Sorry, I can tell you why it's produced in the tip. It was just an observation we could do with this microscopy, and it's really hard to do because you have to grow these fungi on these slides for a microscopy, and yeah, stain them really fast and cover them with resins. And yeah, but we did this experiment to show that it's really produced by the mycelium and not an artifact through the extraction process, and to show that's like it's in a live mycelium present, and yeah.
But what we can say with confidence is that because these compounds that stop the degradation process are found concentrated in growing hyphal tips, that perhaps this fits in with the hypothesis this chemical is for interacting with other organisms. Would you say that's true?
But then it, I would assume that it's going to secrete it if it's for interaction with other. So at least there, I'm not aware that there's found beta covalents out of the fruiting or the mycelium. But yeah, you're right. There's high amounts found in mycelium or in this stage of mycelium.
Does anyone else have questions they want to add?
Well, I'm kind of just wondering if you have any thoughts on the feasibility of this kind of a system being implemented in Oregon. I mean, that's what we're kind of contemplating here. If you have any thoughts on
which system?
Well, biosynthesis, generally speaking, just if you have any thoughts on whether that's a feasible way to go about it. Another path we're considering, which it sounds like we're going to pursue, is extraction from mushrooms. So I'm just curious to, if you have any thoughts on the feasibility of one pathway versus the other.
So in the future, there will be definitely two approaches. So one will be just the pure synthetic compound, which will be, I think, in some capsules, or and then the other one will be also the natural whole product. I think at least that's the development I also can observe with other products. For example, with cannabis, if you see, you can give the THC as a single compound, but as a whole product, it's much more effective, because there are a lot of compounds which they are for sense, for a reason, and also have. So there's effect from THC for sure alone. But if you give, for example, the whole extract, the effects are much better, much more. So the CBD cures maybe some paranoia and stuff like that. So just as an example, and we need to. So I think there will be some people who wants just the pure compound, more in the pharmaceutical approach. It's definitely works. A lot of studies show that, but maybe also the whole product has an influence on really the effects you can observe on that. Maybe it's the reason for the different. For example, if people don't tell that's more colorful trips, that's more mindful trips, stuff like that. And so I think there will be two divisions of people. One for the science approach or the synthetic compound, and the others for the whole product. In the extract could be also whole natural extract.
Well, I'm kind of leaving out synthetic entirely. I mean, my understanding is producing from fungal hyphae in a bioreactor versus producing mushrooms and then either utilizing the whole mushroom or some extract of those mushrooms. I mean, in terms of the expense and complexity of setting up a facility to produce psilocybin through this biosynthesis versus, do you have any thoughts on the cost and complexity? Because, that's kind of some of the complexities that we're having to consider here.
So fruiting bodies can be grown for, I'm not sure, $5. So that's hard to beat. But so the in vitro synthesis is definitely the more costly part. But it's much more easier to work, for example, with this GMP I told you about. So for example, if you would be a pharmaceutical industry, I think they would much more go forward with this in vitro approach, make a good standardized method in their lab, and then do always the same in the future. So it takes a lot of money in front, but in the end, it can save you a lot of money. So there are also new approaches outside which combine like chemical synthesis together with in vitro synthesis, which are really effective now. And there has also several advantages. So you don't need to use heavy metals for the organic synthesis, stuff like that. What maybe also the people like. So if you use the enzymes out of the fruiting bodies, you do it with the natural way the mushrooms do it. And I think that's also a way the people would like it. It's not like it's made out of reagent tube. It's made from the enzymes of the mushrooms. And yeah, it's the much more expensive step, but you get in cleaner product. So that's definitely more the approach for the pharmaceutical industry, which has a lot of money to standardize these before. And they definitely can use in vitro assays or just use some enzymes to maybe speed up their synthesis they already have.
Thank you.
This kinase, for example, is really interesting for people. This phosphorylation step is a hard step to do with organic synthesis.
I should call them fruiting bodies so that I sound more sophisticated.
So does this help you, the answer?
Yeah, no, it's helpful. Thank you. Mhm.
Yeah, it is very helpful to hear the pros and cons and consider these various psilocybin product options with respect to cost on the front end, efficiency on the back end, how those relate to who can be involved, barriers to entry, and product safety, purity of product. If I heard you correctly, for once you're set up, which is a large investment, right? And you need fairly sophisticated technical SOPs to run this equipment. But once you're set up, you can make a gram of purified psilocybin for around 2,000 US dollars. Is that right?
So that was the calculation from the chemical synthesis protocol, the old Hoffman protocol. So it was hard to find numbers in the internet for these compounds. And it's also hard for me to measure this. I can tell you how much these compounds or substrates cost, but it's for me hard to measure my working time or something like that in a final compound, because I also did not achieve big amounts. If you go for bigger amounts, everything gonna be cheaper. So this 2,000 US dollar was for the chemical synthesis from the old Hoffman protocol, and just to have some number around. And in today, there are also much more cheaper synthesis outside there just to be.
But just as a frame of, that's very helpful, thank you. But just as a frame of reference, so 1 gram of purified psilocybin, and then translating that back into dosages that patients are receiving in clinical trials right now, and traditional dosages which have been based on dry weight of whole mushrooms, which again are less than 1 to 4% roughly dry weight. How many people could you treat with 1 gram of psilocybin? Hundreds, thousands?
No, so yeah, it depends which effects you want to achieve. So microdoses would be like 5 milligrams, and full doses around 25, 30 milligrams. So with 100 milligrams for people, so with 1 gram, you get 40 people with full dose experience.
And how long would that take you to produce in the lab? A week, a month, a year?
If you have this setup, it just takes 2 hours on the bench. But you have to grow the bacteria before, and so I can give you a little bit insights into that. So just grow like your bacteria in big flask or in a bioreactor. You harvest the cells, and then you break them up, and you can purify the enzymes from your strain which you made for producing them, and work them with them in the lab. And you can also store them in the freezer and work some days after with them. And then it's really easy. You just have to use a reaction tube, and if you have the right buffers and the coenzymes, just pipe them together, wait for some time, leave it on your bench, and then the enzymes make all the work for you. And at the end, it's a really clean product. And another way, what I wanted to also to bring in is the in vivo methods. You can also put all these enzyme into one organism. My colleague also has done this with for us, but for example, aspergillus. So it's now a magic mold, and so you can also produce in bioreactors the compounds of interest, and that's also a really cheap way for producing this. But it's also a much dirtier extract. So at the end, if you want to have the cleanest product, this in vitro synthesis would be the goal, or at least you have to take more efforts to clean your product at the end. That's also totally possible.
Got it. So in this vein of comparing and contrasting biosynthetic methods or synthetic methods, I wonder if you could speak to any concerns you have or don't have about using model organisms like yeast or bacteria to produce these compounds. I'm envisioning a scenario whereby, like, what kinds of measures would you need to take to make sure that, for example, bacteria producing psilocybin don't get into public drinking water, something like this?
Yeah, so for example, for the in vitro, that's no problem because you have just some bacteria which are producing some enzyme. But if you're working in vivo, that's definitely could be a problem if you're using Saccharomyces cerevisiae or something like that. It's or E. coli, which is would be love to be in your drainage system and live there. That's definitely a big concern, and that's also the reason why we only are able to give them away for people which have the same permission to work with these substances. So to be clear, we have in the lab the permission to work with these substances and also a lot of substances, and it's also a lot of work to document these. And so there needs to be high standards then. So if that's like a genetic modified organism, and then it's would be basically, I'm not sure if it's already S2, but something like that. So yeah, you would fit in all these requirements you have to have in your normal genetic lab, which is S2, and it's hard to get and hard to achieve. So mostly universities does this, and you need a lot of expertise. And with this expertise also, there comes the responsibility of taking care not to be drain this away, to autoclave all the waste. But I would definitely bring this up into higher security levels, as it is as a modified organism. It's minimum S1, but you can think about putting in to S2 because it's an alkaloid which is produced, which going to be harmful in higher concentrations, something like that. So it's definitely, I think, in a higher security level, somebody classify it, and then there's so many restrictions working with that.
Yeah, thank you. Very fascinating discussion here. Are there other questions from subcommittee or from board? I also want to remind all attendees, if you have public comment, please indicate to Nick Riley via chat. We're going to collect those and take those at the end here.
I think I have one more, and that is, have you or anyone you know had the opportunity to characterize the chemical profile of fresh, undried mushrooms versus dried fungal material? Is there a difference in that drying process that occurs that is significant?
So that's so the extraction process is really important, and the biggest thing to concern about is that it needs to be water free. And so the reason I showed you are the two enzymes which are working against you. So if you fresh mushrooms, which are, you need to chop them, you get immediately this blue color, and at the end, if you work with them with fresh mushrooms in the lab, you have just a blue slurry at the end. And so basically it's not possible to analyze it, or at least it's really hard then to analyze it, because everything of the compound gets be polymerized. Your HPLC will hate you for that, and So actually, we are developed like really this freeze drying method before, and then this methanol extraction as a first step. That's the best thing you can do, or at least we found out. Or yeah, maybe there are other ways which are better, but and that's what we wanted and what we used. And then in the extract, you just find psilocybin, no psilocybin, and because the enzymes could not work, so you can dry it in a frozen way. They cannot work, so that's is the best. And then if you have them really dry and store them, then the psilocybin as the stable compounds will also be stable over a long time.
That makes sense. Thank you.
I have a few questions about the kits. I love this idea for the kits. It's just so cool. So my first question is, what do you see as the need for other diagnostic development? Obviously, rapid visual color change style kits that can be deployed easily without technical equipment are very helpful. Do you see other large gaps in the field where diagnostic development needs to happen?
Definitely. I think that's the reason is because all these compounds are illegal, and scientists were not able to work with them in the lab the last decades. And now this opens a little bit up, and it's definitely important. So we're doing different work with that, and some of them is all this is already this harm reduction work. So if you think about as these fruiting bodies can, or yeah, as this the difference can be, it's really important to test them before. And normal people don't have access to a lab, HPLC or stuff like that. And in Germany, for example, it's there are no official institutes where you can go and bring your sample. And what we do with these colorimetric test kits, it's a new level, because the consumer itself brings it into solution, and the colored compound is not illegal anymore. And as a drug checker, for example, you can now analyze the colored compound. So in Germany, at least, we can do at the first time here, then this work with this analysis directly yeah, on the place. It's really mobile. And it's also legal for the people who are analyzing it, because they don't have contact to the real compound, because they only have contact to the colored compound and can analyze them. And yeah, we develop also these test kits for other substances, which we thought have a big need. We already developed it, for example, for LSD and for MDMA, and included on the MDMA also different compounds which could be inside as a filler and stuff like that. So yeah, I definitely see a big need there, and I think there could be an easy tool. And now with Corona, everyone already did a test at home, and it's not more complicated than a Corona test. So everyone can do it at their self at home, or official authorities like these. I told you that drug prevention centers can do direct analysis at festivals, parties, and stuff like that, and prevent a lot of harm, which could be.
Definitely. To expand on that, I wonder if you could discuss some of the scenarios that you found with the kits regarding the quality of mushrooms that are circulating about. You know, are they diluted? Are they contaminated? Are they just mushrooms with other psychoactive compounds impregnated into the tissues? Like, what kinds of things are you able to see, or do you see that are available right now that maybe are on the less safe side?
So at least the interesting results were totally unexpected variants of the contents. So if you think about that, like a stem or a cap could have different alkaloids contents inside. So the people were really so you have to, if you have really a good dosage, you need to grind up a bigger amount of the fruiting bodies. Like, for example, you grind up 5 grams or 10 grams. And for the test, you just need like 150 milligrams. You analyze them, and then you need, you really know how much is inside in your whole biomass. And this is the best and the most accurate way to actually have an accurate dose. And if you don't do this, the results will be, so you will get totally different results, and this belongs to the diversity, and also diversity in harvesting points, fruit stems, like that. So the people really liked and were happy to can tell before which kind of experience they want to have, or even they want to have always the same experience, so they can adapt their biomass based on the analysis they took. And it gives also a little bit of security, which helps in the mind, that you don't take, like, if you harvest white mushrooms and you don't know how much is inside, what will happen. That's from the psychological view also really nice.
Thank you. I'd like to solicit a last few set of questions from our subcommittee and board, and also encourage everybody, general public, to submit your comments in comment form, please, to Nick Riley. So rather than asking questions, unfortunately, we don't have time for Q&A with Dr. Bly between everybody, but we will take comments. If you please submit your comments to Nick Riley via chat in the next few minutes. We're going to take a last few set of question and answers here, and then we'll transition to public comments.
Yeah, hi, it's David Hart, and thank you for your presentation. I'm sorry I missed the first half. Just to follow up on that last question, did you find wide variation when you did testing? And
Yeah.
what kind of variation did you see?
So I brought in the presentation the example, for example, with these local Psilocybin cyanescens. And in our town, it had like 1%. And the next town, 50 kilometers away, the same species, same looking, has like over 2.1%. So it's like the double of the amount grown in the same week, same strain. So, and if you go outside and thinking, oh, that's Psilocybin cyanescens, nice, let's take 2 grams. It could be having the double the amount you really wanted. And to take this into numbers, if you want to have like 25 or 30 milligrams, as we thought about, and then you get like 60 milligrams, that's a totally different experience you're going to have then in the end. And that's the difference from maybe a little bit colorful, mindful trip to totally degrading.
That was for wild mushrooms, correct?
And
how about for cultivation? Would you see those types of variations with a standardized cultivation?
So there's a variation between different strains. So if I grow different strains at the same time, there were always strains which had higher amounts, and then there were strains which had lower amounts. But even if I standardize the cultivation so I always use the same grains and the same methods, I found different alkaloid contents, and I would bring up the same range, like they can have double the amount, or at least somehow. So you can find like 1.2 sometimes it has 1.8 or even 2%. But still at the end, if you grow them at the same time, there are some which are stronger and weaker somehow. Does this help you?
Yeah, and as you said, you know, the larger, some of that variation can be neutralized if you have a larger batch. Is that correct?
Yeah, and homogenized. That's
right.
the important thing, that you make like a powder and bring it into capsules. And yeah, that's what pharmaceuticals always do with natural product and natural stuff, I think, to get it homogenized. And yeah, that's the only way to really achieve a nice average amount of the alkaloids. But you still have to test it, definitely.
These are really good questions, David. Do you think, it sounded like maybe in your response, Felix, do you think that by standardizing the growth media, the substrates and the other ingredients in the media, does that help to standardize a bit?
So definitely, if you, but what I want to mention is, if you like complex media, then you have always a variation. If you're using different grains, that's totally dependent on the grain, which how it grows there, how many water it has. So you will. It was just an example. So really, a lot of complex factors play into the role, and if you taking these complex factors away and reducing to only just some of them. So the best way would to give the nutrients itself. So you're giving sugar, you're giving vitamins and stuff like that. And that's really then a standardized protocol. It's really hard to say like you're using 1 gram of wheat from the company, this and this. And so I think you know what I want to say.
No, thank you. That's really interesting.
So in the lab, you can make like really standardized recipes, and that's all how we work and the researchers work. And then there's really every compound listed. It's gram really per gram, and you can adjust the pH and control more factors. And but you can also standardize the growing on definitely on soil, but it will still have a little bit variation always, as this is a natural product. So depends on the harvesting point and time and stuff. Really small factors can have a big impact at the end.
That's very helpful.
More questions for Felix? I'm very curious about the quantification of psilocybin, psilocin, and these intermediates. So you said that you need a standard to make your, or like a concentrated product of known structure so that you can make your standard curve to compare your experimental values to. Where does one get such a standard? Do they exist for all these products?
It's really hard to get these standards for the compounds, and I think that's a big problem in the future. And also they are really expensive. And for good luck, there are some companies outside which are really specialized on making standards for forensic laboratories, and there are also already buy some stuff, but be shocked about the price. And yeah, so definitely if you want to quantify with the HPLC, so at first you need the standard for really finding your compound to see which peak is the right one. And then you need really the pure compound as the standard, and you will calculate on this standard. So based how good your standard is, your calculations will be. If you only have like a 90% psilocybin standard, then all your calculations in the past will be based on this 90% purification, pure compound. So to quantify, you always have this pure compound. And even if you did this once in the HPLC and you just change your capillary because it's broke or something, and the volume of your HPLC changes, or you have to wash your column frequently, then all these can change. So you have to do it even several times. So it's not something you just have to do once for a lifetime. You can, if you change something in your HPLC, you have to do it again to be really sure. And also to work with the HPLC and psilocybin is not so easy because it's really polar. And yeah, we did a little bit to develop nice methods here to also get a nice peak separation between all these alkaloids. So you need a baseline separation from in the HPLC really quantify the amounts, because all of the area are going to be calculated, and if there are like 2 people in one, then you cannot calculate the amount behind. Yeah, at least not with the algorithms at the moment. So you need a really good standard behind, and also a good method, a good developed method. So it's not so easy to do this in the lab with the HPLC.
So just to be clear, that's for any psilocybin product, whether it be whole mushrooms, hyphae, synthetics, right?
Yeah, if you analyze it with HPLC, then you need definitely a standard. And that was also the reason why we have now these test kits. So it's really easy to see the people just wanted to see if I have this 1% or this 2% fungi. They don't think about 5% variation. They cannot see with the eye. So yeah, I think the results doesn't need to. So I think I know where your questions go. So for the consumer experience, you don't need like this HPLC result, which is really refined, because if there's 25 milligrams or 26, I think nobody will feel the difference at the end. So I think the products can have definitely some variation, yeah, which is allowed, because the effect will not really change. But if you want to analyze it right, it's a really hard process, and yeah, you need expensive machines for that.
But help me understand. So I agree that 25 versus 26 milligrams may not produce a perceptual, difference in psychedelic experience. But you also said that psilocybin concentration can vary up to 5-fold within one species, and that certainly would create a different intensity of experience, right?
Definitely. Yeah, but what I wanted to say is, like, if you just want to have your experience calculated, then you can do now like a fast testing kit. You will see almost the exact amount, and the small variations at the end you will not notice. But yeah, you're totally right. If you want to be safe before, you have to know the concentration. But at least that's the approach. Every therapeutic approach is based on a concentration, because if you want to do scientific studies, you need a number behind. You cannot say, like, I gave them 5-gram fresh truffles. That's not totally uncomparable in the future, because the next one has like 5-gram different truffles. And so actually, if you want to really use it in a therapeutic setting, there need to be a concentration before to. So yeah, definitely.
So my last question on this topic is it more complicated or challenging to quantify psilocybin content or concentration in complex substances like whole mushroom powders or whole mushrooms compared to more simplified, like a synthetic psilocybin?
So if you analyze a whole fruiting body, you need to do a little bit more cleaning before analyzing this with the HPLC, because you have a lot of, yeah, at first your HPLC don't like it, and you also will then have not these single base peaks I told. And so yeah, but the difference in the lab is not as this big. So it's like 1 or 2 more purification steps, at least for the analyzing. If you have want to have like a final free product, it's much more. So all these tryptamine alkaloids are almost the same. Like I said, it's just a method group difference between them. So they will also appear quite in the same retention time in your analysis. So that's a big problem. And if you're having a nicer product and a cleaner product, then the analysis is much more easier. But yeah, actually with another purification step before, it can be also done in the lab from fruiting bodies. So these methods you develop are really looking, for example, indole alkaloids, and so it doesn't change really much if you're having a lot of other stuff in that, because it will be flush away in your HPLC. You have like your methods specialized for these compounds. For example, the Psilocybin likes really a lot of water, and if you just use a little bit solvent, it will come directly out of the column. And so it comes really in the beginning of your analysis. And yeah, so it has some challenges because at the beginning, all the stuff which is not binding comes also. You have to be sure that it's not in this peak from the beginning, but yeah, this helps you.
Yeah, it does. I mean, to me, it sounds like there's a trade-off, right? The synthetics and the purification steps can be complex and expensive in terms of equipment and protocols and expertise, but they offer a product that's, you know, quicker and easier to quantify and potentially more consistent. And the opposite is also true, that mushrooms are easier and quicker to grow, but because they're more biologically and chemically complex, you need to spend more time and potentially more money testing those products to ensure product safety and consistency for the consumer. That's what I'm hearing, but both are actually quite complicated process.
Yeah, I think it's the same for all of the other compounds always. Then the synthetic approach, the pharmaceutical approach, is always the much more costly approach. Has its advantages, but yeah, definitely there's cheap mushrooms outside, and that no synthesis can deal with this natural $5 grow. So you just need some grains, and this will grow. Yeah. So it's a hard, and that's why I also said there may be two consumers outside then. So maybe like the ones who want this pharmaceutical pill, and the others who more want this natural product approach. I would guess at least the people will be split in these two groups. Like the people who always go more to his doctors and wants a pill from them, they will get from his doctors this psilocybin, just pure compound. And then the other people who are, who want to have the whole experience and maybe informed themselves more and something like that. So there will be a need for both of these variants.
That's a fantastic point. So considering who is going to be getting involved on the manufacturing side, but also from the patient side or the clinic side, how to produce a certifiably safe product. It's possible in both cases, just with different and unique challenges.
Yeah.
Are there any more questions from the subcommittee or the board? If not, we're going to prepare to transition into our public comment period. I have been collecting public comments via the help of Nick Riley, and we're going to take those comments now. I just wanted to remind us all that the public comment period is not structured today as a question and answer. So if you pose a question in your comment, just a gentle reminder, it will be a rhetorical question. So Felix, please don't answer questions if they arise. This is not a discussion, it's a comment period. So our first comment in that case comes from Michelle Wiseman. Michelle, please.
Hi, Felix. Thanks for talking with us today. I have 3 comments. First of all, it's really interesting that the highest content of psilocybin occurs in, I mean, according to that other study you cited, like tidal wave, PE, etc., all these kind of mutant-like forms. From my understanding, those mutant-like forms typically only occur in the first flush of the fungus, and that usually people like do like multiple flushes, and then the subsequent flushes have more like psilocybe cubensis like morphologies. So my comment there is that, you know, it'd be really important if somebody were to use your kit that, you know, there might be, you know, significant differences between the activity of the particular strain tested between flushes. Like perhaps the first flush with these like kind of mutant forms, maybe they're more potent. My second comment is regarding the wood lover fungi. I think you might have mentioned that you can't cultivate those indoors, and I think generally, you know, you can't. I mean, you can, but I think that nobody's really achieved commercial success. So some people have been successful doing that inside, or like in greenhouse facilities or incubation chambers, using kind of like a wood chip based substrate, but nowhere near the success of psilocybe cubensis. And then my third, and I think most important comment is that well, I've thought a lot about field testing of mushrooms. I'm in the Pacific Northwest, and we have a deadly look-alike species that co-occurs with, you know, several of the, especially the wood lover fungi. And I think your kit is really great for differentiating like the gallerinas versus the psilocybe species. But here and in Europe, we also have toxic mushrooms that produce psilocybin that also have other toxic compounds, like species in the genus Inocybe. And so I would be a little concerned about sending someone out who's naive about mushroom identification into the field with your kit. And if somebody found like a bluing mushroom with a brown cap, I tested it and it was a toxic Inocybe species that produces psilocybin, because it would test positive, but then, you know, if somebody would consume it, they would get very ill. So I think it's a really great kit, and I love the fact that you can kind of quantify it too. Just some things to think about.
Thank you very much, Michelle. Those are valuable insights. We've been discussing the importance of using modern diagnostics, including microscopy and DNA sequencing, to differentiate species of fungi. And I think it's worth reminding us all that every species of psychedelic fungus and others that do not contain psilocybin have very visually similar unrelated species that have deadly poisonous toxins that will kill you really quickly in a painful way. So please, if my one message to the public was anything, it was do not consume mushrooms. You don't know what they are. Our next public comment comes from Mike Moon. Mike, please.
Hey there, thanks for having me. I just got a little bit late, so this comment's totally off base. My apologies. One thing just to point out is, you know, trying to get to like a super exact amount of psilocybin is, like kind of Felix was saying, is super challenging. I kind of think about it as sort of like the sweetness of an apple that you might be growing. You know, depending on the day, the branch, when that blossom was pollinated, you might have totally different outcomes. I don't necessarily think a total exact standard or universal standard is necessarily applicable if manufactured or like commercially created mushrooms were done like on a batch scale, and then a simple testing of the batch was done, just annotating like the content of not just the psilocybin but also the psilocin would be useful. You know, typically out in the the current world, when people consume this stuff, you know, there's no numbers or anything associated with, and people start with a small, you know, sample amount, see what the effect is, and then scale up or down as appropriate. So just my 2 cents on getting to a quote unquote standard dose. Currently nothing like that exists, and I don't think it is necessarily a hard requirement to be successful.
Thank you for your comments, Mike. This has been another topic that we've been discussing and considering carefully, and there is quite a bit of concern about quantifying and standardizing doses that are going to patients.
Yeah, the other thing on that is, you know, people have different metabolisms for the substance. So some people, you know, 2 grams of a specific thing would, you know, send them into outer space, and other folks 2 grams would barely even be noticeable. So that's another thing to keep in mind, is that individual tolerances are highly variable.
Yeah, it's David Hart. I know we're not supposed to be commenting on the public comments, but no, there is no research on the pharmacogenomics of psilocybin. And we have to assume there's going to be significant variations among individuals.
Yeah, I just wanted to jump in too, because great point. We do have pharmacogenomics in cannabis, right, that apply to cannabinoids, and that's exactly what we see. Folks can be fast metabolizers of one cannabinoid, slow metabolizers of another. And that makes a significant difference when we're attempting to standardize dosing across a population. And I think the reality is, you know, we just can't, not to the precision I think that we're going to want to achieve. And you know, to the same point, I think to a degree, we're going to have to accept that. And that's why, you know, us being flexible and OHA being flexible around dosing, as we discussed yesterday, will remain pretty important.
I agree. There's so much cool research that, you know, needs to be done to gain insight into all of these issues. Although I'm sort of curious, Rachel, if you know the range of concentrations or sort of dosages of these cannabinoids as they exist. I'm curious how fungi and plants may be similar or different in that way. Is it like orders of magnitude difference, or like what is the, when a product says this range of cannabinoids, what quantifiably, like what is that range?
Yeah, we have information and some data. If I you know, have the time to pull some stuff together, I would be happy to come and share insights.
That'd be great. Yeah.
Yeah.
Like put your insights there. Cool. All right. Our next public comment comes from Will McHugh. Please, Will.
Thank you, Felix, for that incredible presentation. In my opinion, Felix has elevated plant medicine in more ways than a lot of people have on the planet. And my only comment is that he's a rare and precious resource, and I would hope that he would consider coming to Oregon and participating with his family as this process unfolds. And that's all. Thank you.
Thank you, Will. Our next comment comes from Kyle Meyer. Kyle, go ahead.
Yes, hi, thank you. My comments regarding you know, some of the recombinant expression of these relevant psilocybin synthesis genes in yeast and in E. coli, and really just to think how feasibly scalable these current approaches are with, you know, production of these compounds on the order of like a half gram or a gram per liter. Also, given the notion that these the growth media of these bioreactors is going to be inherently aqueous, so, you know, is that going to facilitate additional enzymatic degradation or cause issues at the tail end in terms of using these you know, novelly developed strains of E. coli and yeast to produce psilocybin or psilocin, if, you know, ultimately you know, that's going to be a viable pathway you know, for the state to consider in terms of producing these compounds. And, you know, it seems to me that it's certainly the in vitro route would be massively more favorable. But yeah, if there was any additional comments that, you know, Dr. Bly, you would be able to provide on that front. I'm not saying that that's permissible for the board, but, you know, I would be very interested to hear your thoughts. And thank you for an incredible talk.
Thank you for posing those comments. Nick, do we have any other public comments?
No, I don't think so.
All right. Well, please join me in thanking Dr. Felix Bly. It's been very fascinating chatting with you. We very much appreciate your time and efforts joining us, especially this late in your evening. And yeah, let's wrap there. We'll have a break between this and research.
Thank you.
Thank
Can I
you.
use? Okay.
Thanks, Felix.
Cheers.
Thank you.
Thank you.